Hey microbiology enthusiasts! Today, I'm excited to unlock the mysteries of bacteria with one of the most essential techniques in microbiology, Gram staining, developed way back in 1884 by Hans Christian Gram. Gram staining is still a cornerstone in clinical microbiology. Why? Because it tells us so much about bacterial species just by how they react to a series of stains. Cool, right? So let's dive into the principle behind Gram staining. Essentially, it all comes down to the structure of bacterial cell walls, specifically the peptidoglycan layer. This technique categorizes bacteria into two main groups, gram-positive and gram-negative. Gram-positive bacteria have a thick peptidoglycan layer which holds onto the crystal violet dye. Under the microscope, they appear purple. On the other hand, gram-negative bacteria have a thinner peptidoglycan layer and an outer membrane. They don't retain the crystal violet dye and instead take up the counterstain, safranin, appearing pink or red. Now, let's break down the steps of Gram staining. First up, crystal violet, the primary stain. We flood the bacterial smear with crystal violet, staining all the cells. Both gram-positive and gram-negative bacteria will initially take up this violet stain. Next is iodine, the mordant. Adding iodine forms a complex with the crystal violet, making it too large to escape the bacterial cells. This is especially important for gram-positive bacteria. Then comes the critical step, decolorization with alcohol or acetone. For gram-positive bacteria, that thick peptidoglycan layer retains the diiodine complex, keeping the cells purple. But for gram-negative bacteria, the alcohol dissolves the outer membrane and thins the peptidoglycan layer, washing out the dye. Finally, we use safranin as the counterstain. Gram-negative bacteria, having lost the crystal violet, take up the safranin and appear pink. Gram-positive bacteria remain unaffected and stay purple. So what do the results tell us? Gram-positive bacteria appear purple under the microscope, while gram-negative bacteria appear pink or red. Simple, yet so powerful. The significance of Gram staining can't be overstated. It's a primary step in identifying bacterial pathogens in clinical diagnostics. Plus, it helps determine how these bacteria might respond to antibiotics. Gram-negative bacteria, for instance, are often more resistant because of their outer membrane. Gram staining is fundamental to understanding bacterial characteristics and is crucial in diagnostic microbiology. Thanks for tuning in and happy staining.